Advances in gene technology are transforming disease detection with a new test that promises to be faster and more accessible than PCR. Developed by Professor Eva Goldis and her team, this cutting-edge test uses technology to detect diseases like COVID-19. The innovation could revolutionise diagnostics, making it easier for anyone to identify illnesses quickly and affordably. Professor Eva Goldis joins us now. Professor, welcome. Now, I'm sure that we all remember the COVID testing we had to go through. No one wants to repeat that. So can you tell us a bit about how this test works and how it compares to the more traditional PCR testing?
We are very excited to be showing this to a wider public for probably the first time in a live setting. We developed a technology that is centered around engineered nucleic acids. Nucleic acids are, as everyone knows, a cornerstone of life. There are molecules that contain the genetic code of life. They are specific for each organism, and we are able to detect them by the genetic sequence they contain. Now, what is important are the sequences that we are using are very, very small snippets, and that makes this technology very easy to apply in an end-user setting. So the differences between PCR, the problem with PCR, as you probably all know, is we had this problem during the pandemic. We had to queue in a pathology laboratory, long queues, sick people, and then we had to wait for an answer. And this is because PCR technology, which is an old technology, over 50 years old, is extremely hard to be taken out of the lab. It requires instrumentation, it requires careful management of contamination, and this is why it will probably never be able to make it out to the end user. So the end users were given test strip, the so-called RAT tests, and they were amazing technologies, but their accuracy, as everyone knows, is not as good as we would like it to be. So we are trying to sort of break through those barriers with our novel genetic test that will be applicable in a format that they will look like this. So that you probably recognize. This is actually a cassette from the proper RAT test, but inside is a little paper strip which we made ourselves. So this particular one detects genes of a water pathogen. So that's how it's going to look like. For you and me, it's going to be one of those. You now already know what you do with it. You put your sample into the little test tube, wait. Now, in our case, the wait will be much, much shorter though. We are hoping for those tests to be as rapid as a couple of minutes, under 5 minutes.
Wow. Now is this type of testing, Eva, able to be used on applications other than COVID-19, other diseases?
Absolutely, 100%. So infectious diseases are all caused by specific organisms. They have specific genetic code. We can read that code. It's done by other methods, and we can extract a small piece of this code and use this information to create tests such as ours. What is really important is that this is such a simple process. So you can actually make a test like this in a lab in less than a day. So it's very, very straightforward. I think this is going to be super easy for scaling up of those tests. The development time will be much shorter. The costs are also very, very low. We've done some calculations which indicates that at least in a university laboratory, to make one of these costs less than $1. So we really can hopefully transform a lot of situations where testing is really needed. A significant objective would be maybe to look at public health settings such as airports, quarantine, animal health as well.
Well, it's incredible that you can use it like you are. So let's talk about when we will be able to access those sort of tests. You know, what are the next steps now for commercialising the NanoCircles test?
Look, there is a whole trajectory to it. And we are well advanced on this trajectory. First, you need to protect your innovation by a patent. The patent sits behind it. It's that thick, actually. So then, obviously, we need to have a commercializing partner, and we need to have bridging funding to sort of really enable us to jump over this valley of death, which we believe we have successfully attracted. And then the question is, how do you scale it up? That's a really interesting story. We are living in really interesting times because the COVID pandemic has given everybody the recognition that RNA technology is really an important technology. You probably remember mRNA vaccines. So we will be building on the industrial infrastructure that has been developed for the COVID vaccine, in a sense. So it's going to be exciting, and we can't wait. And because you can actually really detect everything that you want, including, very curiously, environmental impact. So I don't know whether everybody is aware, but we are obviously concerned about the impact of different things on the environment, such as climate change, but also sometimes adverse effect of industry on a particular environment. We are concerned about those native Australian species being threatened and so on and so forth. Many of those species are really hard to find. So how do you know that a particular species of organism, be that a frog, for example, or a koala, is being negatively affected? We currently are going with binoculars and counting koalas. However, you can actually trace koalas by the residues they leave in the environment. They leave a little bit of a feces, maybe a little bit of saliva, and all that contains DNA. And if you have a sensitive methodology, you can actually track down those animals by the traces they leave in the environment. And we hope that this technology will revolutionize that. So there is a broad range of different applications for gene testing.